Thank you. Can you hear me? Yeah? Okay. So I have a very low voice, so. Okay, so so as you know, a key obstacle for vaccine for AIDS is the inability to deliver antigen to the in a sufficient period of time, leading to a weak or transient protection, and sometimes people have to boost many times in order to have immune responses. So in my lab, we were looking at the, this is the vaginal and the cervix here. We're looking at the entry, and we thought, well, you know, when the virus goes in, the primary phase of infection is really characterized by the early and drastic burst of viral replication. Then you have dissemination and then induction of immune response after you go all to the lymph nodes and propagation and so on. So it's actually very fast. And when you look at the acute phase, with a few days, you actually have a lot of viruses. So we thought, what about if we could somehow block the entry of the virus at the site of entry, which means in the mucosal site of entry? And we thought, well, you know, there is a lot of immune responses here and a lot of cells that are actually here, but how can we do to actually stimulate them without, make a vaccine that will be maybe one-shot vaccine or one vaccine at a time, and then long-term immunization and maybe protection. So from that idea, we start to develop different things, and this is a schematic of the vaccine concept. I want to say that the primate is a great model for vaccine concept, for proof of principle. Anyway, so here the idea was, what about if we could use, so this is a cartoon of what you just saw before, if we could use the epithelial stem cells, and specifically the basal layer, which is right here, the basal layer, as a permanent source of antigen or viral antigen. And if we do that, if we could put antigen here, then the progeny, which is the differentiated offspring, will serve as an antigen-producing presenting cells. So it would be very simple, conceptually, on paper, to just deliver the viral protein in the epithelial stem cell layer here, and then wait for the progeny to continually heal the new antigen-producing cells without being actually rejected or eliminated by the immune responses. And then you should have, in theory, an antigen expression for long term. So one of the questions is how we could do that. Well, we could maybe find a promoter that will drive the vaccine to those epithelial stem cells. And then when it's here, then the cells themselves will continue the job. So based on this principle, we send a grant and we get funded, and then it actually is easier on paper than in reality. So it took us a long time, and we're still working on it, but we are pretty slowly excited about it. So there is different promoter which are specific for the epithelial stem cells. So we choose the involucrine promoter. It's differentiated epidermal mucosal keratinocytes. We use it because you actually see it in human, in monkey, and in mice. So we thought that could be a good tool to see the construct work. And then from there, we start to investigate it. Okay, let's see monkey, how we're going to do that. And the idea we had was, what about if we don't use any needle, we just drop in? So we use like birth-feeding tubes, and then we just put the concentration of vaccine. I will go to that in a minute. And we drop it in 1 ml or half an ml, and then we wait. And the monkey is like lay down, and then after that we monitor. And then deception was we got nothing. And then we realized when we start to look closely, and I should know that, but I didn't, because I actually never pay attention on that. But actually there is ovulation in the macaque female, which is very complicated. And what happened is during the ovulation time is when the epithelial cells are very thin, almost to the basal layer, and that would be like here. But when you are at the beginning of the that would be the menses, the end of the cycle. And as the cycle goes up, the epithelial cell got thicker. And then when you have ovulation, you actually have this white, hard, impossible to work with inside in the ovulation. And what happened is if you drop the vaccine here, there's no way the vaccine is going to go to the basal layer. So what we did, we start to do treatment like Depo-Provera and all these things. And what we find out, and then and so on, people have used it to infect before whether you can use that. What we find out is it actually works very well. It clean up totally, so the vaccine is there. The bad news is the cells don't really come back for months. So the effect of the vaccine is not to be there, it's actually that the cell can proliferate back up and then and then the antigen is going up and stimulate the mucosal area. So we thought that's not good, so we tried Depo abrasion and all these things, I passed the detail, and we decided to just follow the cycle. So what we did, we used this with a collaboration. We used endoscopic coloroscopy. We did optimal Korean topical OCT, and we monitor every week the all female to see when there will be the mensum and so on. And what we find is during the mensum, actually it's very close to being very thin, and that's perfect time to actually immunize. And then we wait for no mensum, which is post, which will be maybe the ovulation. It's huge, and you can't really go through that. And then so we monitor the female, and actually they present themselves, they get used to it, and we give treat, and everything is fine. And we wanted to do a non-invasive drop-in, no cut, especially no cut, and then do it. So we did. And then so when we got that right, we thought about doing the vaccine. So we decided to do a single-cycle vaccine, something that will go into the epithelial stem cell and stay there, and then the mother cells will do the job, I guess, sort of say. So, and then we did that, so it was a single-cycle vaccine under the control of an evolution promoter. What we did a little bit of changing to make a single cycle in the LTR and also in the TAR, and then we moved the NEF. We actually later on add the GFP to be able to tag it, to see it. And we also did one with VIF and no VIF, no NEF to make it even more safe, I guess. Okay, so this is very, very contracted, but it's all published. What we did was the chaos was the virus and the vaccine can only do one round of proliferation, I guess. So what we did, we had to actually make liters of it, and then we ended up using a cross flow to actually concentrate to have about from 2 liters, about 10 ml of vaccine. And after a more process, we were able to have the vaccine, and this is all here and it's published. We did the proof of concept on mice, and we saw the tag. We did different sites of immunization drop-in. The skin, we just did this like that, and then just drop it in. And then you could see some green, and then we went to the monkey after that. So this is published, and I'm going to the monkey now. So here, so as a proof of concept, we took female macaque. They were all mammu at the time. We did, at the time, we didn't know what to expect, so we did the intravaginal, rectal, and transcutaneous immunization. Again, this is drop-in only, or on the skin. And then we had the control with no antigen. And we monitored the animal. It was four females for 24 weeks, sacrificed 2, and 2 were sacrificed at 8 weeks post the vaccination. And then we did, the idea was to see if we could see anything, I guess. So one thing very important was, okay, so delta F, radio are 2 delta F that we had from previous study. And here what we did, we tried to compare and see. So this is time of infection, 8 we sacrificed 2, 24 we sacrificed 2. And here we're looking at what we can detect in the plasma or cell-associated. And we saw no cell-associated virus. We had no cell-free virus detected in the plasma. We could not recover in any of the culture. We took PBMC, vaginal cell cultures, nothing. So to us, and there is no visible site. So we thought, either it's in or we lost it. So we keep on going with that. But we're very excited because the delta F, this was a delta F, by the way. We start all the primary experiments using the only delta F, single-cycle delta F GFP. Here to look at those who are mammo positive, looking at CD8s, we sacrifice, as I told you, at 6 weeks or 24 weeks. We could see actually good responses to, this is all gag responses. I don't know if you can see from here, but this is to 1%. So we were very excited to see, well, at least, and this is vaginal cervix and so on. So we could see actually a response specific for, so we knew it was there and it was actually presenting. And we calculate, okay, 4 weeks being the cycle, this will be 8 week 2 cycle, and then here we are already more cycle. So we thought, okay, more cycle, more expression, maybe not. Again, we had no clue. But we're very excited about this. Then we did some in situ, so we have lots of samples that we're just starting, so I just want to show you here. This is one animal that we followed. Those are only biopsies. Animal was sacrificed here. And then Bepard, we GFP, you can see the GFP here. If you look at the merge together, this would be the basal layer. So it looks like it expressed in the right place. Again, when we dropped the vaccine, we were at the level of the basal layer, and it looks like maybe it's working, so we're excited. And then I spoke to François Villinger, And he did this. I don't know if he's back on lunch, but if he is, question has to go to him, I guess. Well, help. He's not here. So anyway, so what he did here, and with Philip also, both of them, I told them, well, I would like to visualize the vaccine. Where is it? So what he did, this is just one animal, and it's like a concept also. We're going to go more now into that, but we wanted to have an idea. We basically follow, so basically the animal is immunized here at what we call day 0, I guess, of the cycle, and then 48 hours after we label with, here we label with a label marker, and we scan with CT PET, it's an immuno PET with an anti-CD120 probe with an isotype. And then what we do, we follow that with PET CT here, and then we look. So this would be the early phase. That would be, I cannot see from here, probably you cannot either. This will be the, I guess, 8th or something like that, just before the ovulation. That's 2nd month, 3rd month. And then here, this would be the 2nd month and the 3rd month. This is the control. This is the bladder, and I was told that you actually have sometimes that problem. But if you can see the virus sign here is clearly into the vaginal and maybe cervix cavity, and you can see in the draining lymph node as well, which is kind of great because we actually don't see into the blood, as I say, and we cannot express it. So we're really excited. And again, this is ongoing. We are following that with more animals, and we're going to be looking at that more seriously. I mean, it was serious, but more. So anyway, so then we went back and so this was done also with male, but I'll show you only the female today because we have no time. So 6 in female, what we did was 8 of them was vaccine, the other one was controlled. Because of lack of time, we decided to do, let's try to challenge after 12 weeks, which is 3 months, 3 cycles, or 6 months, 6 cycles, and 4 and 4. And we thought, maybe we don't have enough immune responses, but let's just try. The dose we put is 500 nanogram P27 equivalent. I forgot to tell you this. Yeah, it's a 239 SIV, 239 single cycle and so on. Anyway, we use bird feeding, and the animal are monitored overnight, over time, for 3 to more months. And so this was done by PAM. We looked at the immune responses. First of all, the antibody immune in the secretion. And then here, we did the secretion at every 2 weeks, I guess, which was actually, when I think of it, not great because if you imagine this is the beginning of the cycle, 2 weeks would be the ovulation. So, next time we do it, we're going to do it like every week and not like that. Anyway, but you can see clearly good vaginal, this is IgA, envelope IgA responses. The open are the 12-week and the 24 weeks are the other one. I guess it's limited amount of animal, but it worked well. We also were able to detect the vaginal, this is the question, IgG, envelope IgG response. We could not see anything in the blood, which makes sense because we could not see anything most of the time. So it's a little bit, it's a little bit various depending on the cycle. We had some problem to collect the secretion. Now we nail it. So the next study is going to be beautiful. But at this time, it was kind of a little bit empiric. Just to finish here, I will say that so we developed that vaccine against involucrin promoter, single cycle delta, MEF, GFP. It's a 239 backbone. We were able to drive the expression to the differentiated epithelial stem cells, nothing in the blood so far. We induce a strong IgA, IgG response, again, the envelope. We cannot see any gag, anything, just envelope. And also, I can show you, but we have also a good locally, only in the mucosal IgA, IgG, CD4, CD8 that express alpha-4, beta-7, only in the mucosal. It was safe so far. We didn't have any problem with the animals. I will show you that in a minute, but we actually followed some animals for 3 years, and we had no side effects, so we are pretty happy on that. So the next step was to challenge, and we decided to do the vaginal challenge. We used a 239 stock. It was a 2,500 PCID50 repeated dose, and we thought we'll do it until all the control got infected. It turned out that they got infected pretty fast, so we ended up continuing until all the vaccines was infected. So maybe it was not great, but we did that. So it was 2 groups again. So that was 3 months later challenge and 6 months later challenge. That's about all I want to say. And this is a typical 2-time week up to 8 weeks where I actually did all that. Although the vaccine, the control were already infected. I want to show you this little bit. So if you look in that panel here, this is the first group of animals. And what's interesting to me is that we have maybe 1 log differences. Again, it's the minimum amount of animals, but you can see clearly a differences even after only 3 months here of vaccine. You can also already see a trend here. And when you look, this is all together. The open will be the animal that were 3 months vaccinated. Those are the 6 months vaccinated or 24 weeks here. And you can see some of them seem to really do very well. And if you look at the long term, so we are super excited. We thought we'll not have that at all because it was so short, but it works. If you look here, this in red, the color doesn't make sense, but the red is the 6-month vaccine and challenge, and the green being the control. And you can see clearly you have an advantage. Also, you have I think that we could see a delay of infection, but they got infected, yes. But again, all the control were infected way before that. We kind of push it. The blue here is the one that were vaccinated for only 3 months, and you can see, it's maybe maybe I don't know, there's no significance, but here is more. So we're thinking maybe we should have waited more. So that's the next time we're going to be waiting longer before 2 challenge, assuming that the immune response will be maintained, which we don't know if we need to boost or not. In theory, we should not need to boost, but again, I don't know. So this is some of the immune responses, just to show you that this is, of course, after challenge. So we have a lot of immune responses, a good T-cell response, the response, and blue being the 12 weeks and 24 weeks or 3 months, 6 months. Nice, mostly actually were 1 function, and on the we looked at TNF, interferon, IL-2, and CD107A here. One thing I want to tell, so look at the antibody. This time, of course, we have induction of systemic antibody response, IgG response, again, the envelope. This is a 12 weeks challenge and 24 weeks or 6-month challenge. It's possible that we have an amnestic response, although it's complicated because, so the thing is, I didn't want to take the secretion during the infection because I was so, I thought, I don't want to touch it to mess up with this. We're just going to infect and move away. So we maybe missed it. I should have maybe done it half the week, like 4 days after, and then challenge 4 days after or whatever. I didn't do that. So it's hard to say here because we don't actually have all the time points. And when I call after infection, depending on the animal, it could be after 7 infection, 7 transmission, and they got infected, or within the first few weeks. So it's a little bit misleading, but the thing is we see nice responses. Overall, this is just cutting fast to the data. We see this is a 12 weeks vaccine, so 3-month vaccine challenge. We see inverse association. This is the IgA here. This is at the 4 weeks after a viral load, and here 6 weeks after. Well, we see, again, there's minimum animal number, but you see an inverse association between both. We see IgA. When we look at the 24th, so 6 months post vaccine we challenge, the IgA and the IgG here, and you can see you have a inverse association. We're looking at IgA here. This is a 6-week, and this is 4 weeks after the viral load. However, in the serum, you see the positive correlation, which makes sense. And that's where we have a strong T-cell response as well. And one thing I will tell you, so this is a 24 weeks, so 6 months stimulation, and then vaccination and we challenge. So this is my 4 animals, my 4 animals. They went down, and then this is limit of detection. So they are really bumps. We look really down, but it's really very low, almost negative, but we were able to detect it. And here we thought, well, let's see if we deplete the CD8. I had to stop. We were running out of money. So we depleted the CD8, and boom, we were all surprised. So, but the good news is, so yes, they rebounded, all the virus rebounded, but this is an anti-CD8, the way everybody do it. What we find was that They actually went to undetectable within 8-10 weeks after that. And then we maintained them another 6 months. We had no bleep, nothing. And then we had to sacrifice. So the whole animal are frozen and all over the place, brain including. So to look what we're going to be looking. But we're very, and remember, this is only one drop of the vaccine that says we don't do anything, we just drop it in. So we are so excited about all this so far. So conclusion of all this will be that we have, we have different significant control of viremia. I will say, yes, they are infected, but again, we kind of push the system, and this is very minimum animals. One thing I want to tell you, I had 2 MAMU and 2 MAMU negative and positive in all my, they were all the B17 negative. So what was interesting is that it doesn't really matter, MAMU or not, at all. So, so that's good, we don't have to worry about this. We find mucosal exposure demonstrates a positive correlation between the generation of mucosal and systemic T cells, also inverse relations with the viremia, with the IgE and IgG. We also see, of course, an increase of T cell response. Of course, when the virus goes down, we see nothing anymore. It's very interesting. It's like being in treatment, although we don't treat anymore. So it's really going down. So we're super excited about this. And that's pretty much it. We have a dramatic increase of viremia coincidence with also B cells, and we had a lot of NK. So we are going to be looking at very closely at what kind of cells actually are there. What actually, did we cure the virus? Where was the virus? All these questions that are still pending. But the key thing is we can really use the epithelial stem cells as a delivery system for antigen, and this could be adapted to anything, not just for HIV. It actually may work better for other vaccines, other virus. So, but what we can say is so far we have a rapid and durable control, and we have also a very good mucosal responses, which is very exciting to us. And I would like to thank you, everybody. I don't have François Villinger or Philippe, but they're not in the room. So, but anyway, Pam did all the antibody, and she's now working with the male. Philippe Blancourt was in the rendezvous and helped me to set up all these constructs. This is a team at the New England Primate that did all the assay with the coloscopy and everything, and this is my team over the year. This took us a while to get to today. Thank you. Yes?
Mario, those are very powerful CD8 responses that your high magnitude responses. Why don't they eliminate the epithelial cells that have the vaccine in them?
I don't know. So when we maybe they have it. We didn't really look at it much. So I don't know. We don't see it much, but I don't think that the next one we're sending, we're looking more to visualize where the vaccine is.
Wasn't that an image where you had those?
Yes, but this was like we did one female after 3 months, and we got those images with François Villinger, right? And you're talking about the
IHC, or immunofluorescence.
Oh, immunofluorescence, yes. So you see it.
Yeah.
But you know, we sort of follow, and we know exactly when it will be expressed at that level. So we took some biopsy at that point. So we are still working on that. This is just one of the first image we got. But so it's there, and then I think it's because of the again, it's expressed, and then probably a cell. You know, after the ovulation, I think the cell desquam, and I think everything sort of goes away. And then when the cycle starts again, you have the epithelial cells, mother cells that go, and then everything is expressed again. I think that's why we see it more in the beginning of the cycle, and we don't see after the ovulation much. So it's kind of interesting. So it's like that was the idea, to try to boost the immune response naturally using the female normal ovulation. But it's complicated because on paper it looked good, but actually we could not really find the right time to get biopsy. And when we get biopsy, we have so little amount of cells, and we're always afraid. I was afraid always to cut too much, and you know, anyway. So it's a proof of concept, but apparently it worked. So we're all excited.
Brigitte Sanders, NIAID. So I remember that Neil Almond from NIBSC in England, he also had a single cycle vaccine. Are you aware of those data? And I don't think the vaccine did very much, but I don't think it was given intravaginally. I think it was given systemically.
Yeah, it was systemically, I think, yeah.
You're familiar with those data?
Yeah, I saw some of those data, yeah.
How are the two vaccines different? I mean, they have different promoters and...
Yeah, so I think we were able to control much more the entry that he has. I mean, this is also a minimum amount of animals. So we are now going with larger amounts. Actually, in men also it worked well. So I think because it was systemic, we actually. So in other words, when you get infection, then it's controlled. But the strategy here was to try to avoid infection, I guess. I think, but when you go systemically, then the virus has to go in order to be controlled. So that was the difference we had. And I don't know what happened after these vaccines.
Yeah, I think it's published. I have to check the publications again.
Okay, I have to check it. It was some time ago.
More questions?
Questions of
do you know any specificity? Do you know the specificity of the IgA and the IgG?
Okay, so I don't, because I wanted to look at the gag response, because I thought there was maybe some gag antibody. And what we did, we really used most of the resource we have on the secretion. So now we have to go back and do that more. I mean, it's very different. Yeah. We really didn't expect this result, so